reflections. So please, would you take a moment to go to the daily reflections page and fill in your daily reflections. We're waiting for one more panelist to join us. Hopefully he'll be on in one moment. These daily reflections are very important for us. They help us figure out what we've done well and what we've done badly. I see our last panelist has arrived. Good morning, Arvind.
Hi, Eric. Sukbin.
So,
hi, Sean.
Hi.
Hello. So the students are going to take a minute or two to fill in their daily reflections. And while they're doing that, why don't we start by having you briefly introduce yourselves. Sukbin, would you like to begin?
Yes. Hi, everybody. I'm Sukbin Lim from NIU Shanghai, and I'm investigating the network models of learning and memory.
And Sean, would you introduce yourself?
Sure. I think many of you have seen me before, and I'm Sean. I'm Sean Escola. I'm an assistant professor at Columbia University in New York, and I'm also one of the organizers of the Neuromagic Academy. I'm excited to be involved in this panel today because a lot of the recent work in my lab has revolved around using complex dynamical models to understand the production of motor function, especially thinking about the interactions between motor cortex and thalamus, and we model those interactions and think about their consequences in the production of complex behavior.
Thank you, Sean. And Arvind, our returning guest. I think you're the leaderboard panelist this week for Asia time zone. So,
am I? Okay. Well, I'm here only for 2 days, yesterday and today. So you saw me yesterday. For those who were not here, so my name is Arvind Kumar. I'm based in Stockholm, and in my group, we are studying dynamics of large-scale networks. Some of the topics that you did today in the tutorial, that's what we work on. And I'm also partly responsible for setting up the day's curriculum. So if you didn't like it, I'm responsible for it.
Well, now you know.
And if they didn't like it, then you're not responsible for it. Right. Is that the?
It's always good. Yeah, it's always good to know who to praise and blame. My name is Eric DeWitt. I'm a research associate here in Lisbon, Portugal, at the Champalamo Research Center. And I'm also one of the organizers of Neuromatch Academy. It's my pleasure to host this morning. And so I guess Sean and I being organizers, you can also blame us if there's something terrible today. All right, so let's begin. I hope you've had a chance to fill in your daily reflections. And a reminder, please set your chat to panelists and attendees so we can all see the messages, both all of the participants on the panel and all of you. So let's start off by looking to see what questions people have asked. Please remember to ask questions in the Q&A panel, and please upvote questions you find interesting. Seems we have so far one exciting question, or not two exciting questions. Hopefully all of you found the tutorials interesting today. I guess what I would like to do while we're waiting for a few more people to post questions is ask Arvind to make a brief connection between yesterday's spiking models and today's dynamic systems models. So we talked a bit yesterday during the question and answer about this sort of relationship between making choices about abstracting away details. And clearly one of the things we've done between yesterday and today is we've abstracted away a number of details. Do you want to make a brief comment on that?
Let's see if it can be brief. So yesterday we worked largely with spiking models, and today's intro also started with spiking neurons, putting them into a network context. And then Nicolas Brunel, what he did was that he narrowed it down and he brought it down to a single rate equation, that how can we reduce the complexity of a large population of neurons to a manageable number of equations. And in that case, it was one or maybe two equations. This is, I think, a good lesson in modeling as well, that as you scale up the system, you can't handle all the complexity. So you need to leave out the detail and keep most relevant properties of the lower level. So today in the tutorials and also in the lectures, what has been done is that we forgot about that we have neurons, but we remembered that we have two kinds of synapses. One is excitatory and other is inhibitory. And since we are interested in a network by definition arises because you have connected the neurons. Therefore, we are interested in effective excitation and inhibition. And what do they mean to the network? Now, all kind of biological detail is either thrown away or compressed into very simple variables. For instance, the complexity of neuron in terms of its spiking behavior, that has been thrown away. And we say that a neuron is described by its firing rate, which is the number of spikes it generates when you give it some input. This is something perhaps you can remember. Yesterday, you did calculate the FI curve of a neuron, meaning you inject some amount of current and you count number of spikes this neuron elicits. So we just borrowed that aspect of the neuron from yesterday. Other thing we remember from yesterday is that we had two types of synapses, excitatory and inhibitory. And it turns out that this is a very important component of the network. It determines its stability, dynamical properties, and so on. So we keep that. But we also keep that in a very simplified way. We keep that in the form of another number called W, which will have a positive or negative sign, which reflects excitation and inhibition. It turns out when you take just these very simple properties and you put them together, very interesting phenomenon arises. Some of it we can directly relate to the brain. For instance, we can relate to response properties. Perhaps you remember it from Ken's lecture, where he talks about how you can explain using these very simplified models what happens when you present different kind of gratings to the animals and whether you can explain their firing rate behavior. You can also take it upwards and talk about large-scale behavior. Yesterday, there were some questions about brain diseases. So you can relate the stability of the network to the health of the brain. And I guess Sean is here. That's great. So Sean knows much more about this. So perhaps he will bring some information on that.
Yeah.
So that's, I think, the big picture. And this was the reason to narrow it down to
no,
this
I think that was a great bridging. I actually, we've got a question here, which is actually talking specifically about this from Joshua Arnold, asking about this trade-off. And one of the things that clearly we lose is the fine temporal dynamics. So we don't have spike times anymore. Sukbin, would you like to ask, like, what is the reason for allowing this trade-off? What are the advantages that we get from going to some model where we can do more analytic analyses on the dynamics? While obviously giving up on that more precise, fine detail, which we think is potentially important in the biology.
For me, why I take the this rate model in the first place is the simplicity, and then it's easier to understand the mechanistic picture. So by simplifying it, like for instance, in the working memory, and then to reproduce the persistent activity, right? So there can be several mechanisms. I mean, there is many debate, like whether it is based on the attractor dynamics or the feedforward dynamics, right? So in the dynamical aspects, like what is the critical requirements in the connectivity wise or the synaptic dynamics wise, we can reveal such a question in the first place using the rate model. And then like if I need the additional, like for instance, if the correlation matters, if I think in the working memory, for instance, not only it is encoded in the persistent activity, the level of the persistent activity or the pattern of the persistent activity, but if I think, I mean, if the experiment suggests that the correlation, for instance, is important, like then I will try to see the spiking network to capture such a spiking, I mean, the relation, spike time relation between the neurons as well. So my approach is increasing the complexity, I mean, that depending on the experimental demands and also the mechanistic question I have.
Sean, do you want to add anything to those?
Yeah, I mean, I think about this a lot, and I think that there is reason to have some concern that we are, going to such a simple mode that we might be throwing away very important details of the neurobiology. I mean, we talk often about losing temporal resolution by going to rate models, but we also lose some spatial resolution, right? We don't think about conductance delays and so many things that go into that exist in real neurons. But the advantage, I think, is exactly what I agree with Arvind and Sukbin is in my work, we often look at the linearization of these networks to try to get some insight. And in the rate setting, that's so much easier to do because we have sort of a simple understanding of how that's possible. And there's so much insight that you can get in that setting. And there's a lot of experimental data. I'm thinking of things like out of Mark Churchland's lab and Kristen Shenoy's lab that support the view that linear dynamics are not too far off the base, at least in some circuits in the motor system. And so I think the field has agreed, maybe I shouldn't say that for everyone, but at least people that have used this approach have come to the conclusion that right now there's a lot of things we can learn from these models. And certainly there's an incredible amount of computational power even in these very simple models. And we should put in some effort to try to understand what this computational power is. And then that puts us in a place where we might be able to ask the questions, okay, well, what's missing? And start to think about whether or not those things come from details that we're neglecting. That's kind of my
yeah, no, I think that was
view.
I think this has given us a good perspective that we gain something by going to these simpler models. So the ability to scale up and look at, say, a whole working memory network and its dynamics as a network, or to be able to use analytic tools. So I think that was one of the things I was curious to point out. When we work with something like the Hodgkin-Huxley model with actual models of ion channel dynamics, and maybe we model a full compartmental model like we were discussing yesterday with active dendrites, we get to capture the computations in that single neuron. But at the same time, the complexity of the model is such that we can't use a different set of tools. And for some questions, I am still having video problems. For some questions, so back to old school video. So for some questions, the analytic tools really give us a lot of power that we don't have when we're using these more complex models where we have to fully simulate the dynamics. So, when you can use the tools of linearization around, some point in the system to look at its local dynamics, you can then basically use some mathematical tools that are much more powerful in some ways. And I guess, as we've been talking about in Neuromatch Academy since the week one, it's a question of aligning that to the scientific question you have. Is it something that's appropriate and necessary for your question?
Can I also ask a question, regarding this? So yeah, analytical perspective and then the modeling, the mechanism, I choose this simpler method, but it raised me a question. Can I ask a question, or I'm rather?
Sure.
Okay. Yes. Then in the machine learning or the deep learning literature, the people, are using the rate model. If I made the analogy to the rate versus a spiking network model, right? Then why do you think of course there is the approach nowadays, they try to implement this machine learning, deep learning in the spiking network, but then what would be the advantage? Although, in the machine learning literature, deep learning, there is no, the analysis is difficult and it's not trying to understand the mechanism, right? But still they are utilizing the rate model rather than this spiking network. The energy and those approach, the people prefer this spiking network, but also the readout wise or the modifying the connectivity wise, is there any benefit of having the rate model versus the over the spiking network?
Well, I think that maybe this is not a direction we necessarily want to go in this conversation, but in machine learning, the tools and data sets that they have to benchmark their models are not necessarily a rich enough set, I think, to be able to answer this question. So yes, you know, the mileage that has been achieved out of rate models alone doesn't seem to show any weakness with respect to spiking models. But I think that you could easily dream up a scenario where there are tasks that the human brain solves for which spike, for example, spike timing becomes very important. And I mean, we know of some of these, right? We know of some of these. There is a coincident spike timing in the auditory system that's necessary for being able to locate sound sources in the three-dimensional world that we live in. And so that this is so that there are features of our processing that does take advantage of a nonlinear, complex, ion channel driven properties of neurons. But I think the question that we are trying to answer as a field is, are those the rare cases? Are those the exceptions? Or are those more general? And I think that there's some reason to think that those are the exceptions rather than the rule.
Yeah, and speaking, from the side of thinking about how we often do our analysis of neural data, when we start looking at, neural activity using electrophysiology, calcium imaging, the first approach we often use is to calculate rates, right? We think about the system in terms of the rate properties, and we've been using that as our starting point for lots of both neural analysis and neural modeling, and it's gotten us very far. But as Sean pointed out, then you will hit moments where that will no longer be good enough. I'd like to switch to a question about what we can actually do when we start working with these models which have analytic properties. So there's a question here about what generates stable fixed points and unstable fixed points, and what is it in the biology that would create this, and why is it important for us to think about it? So Arvind, do you want to begin?
So what generates fixed points is the connections, because that's the only thing that we put in this model. If we just kept one neuron and looked into its dynamics in terms of fixed points, then everything is a fixed point, because there's no dynamics as such, if you wish, because there are no interactions. It's just feed forward. The moment you wire it up, meaning the neuron is able to see its activity or its activity through others' activity, then it starts to dynamically change. And then we bring up the concept of fixed point, that whether the system ever reaches a stable state or a steady state. So connections are important. In these tutorials, obviously the nature of the fixed points and the number of fixed points are determined by the choice of connection weights you make. If you have a more complex system, then obviously you will have to go into more properties of these matrices. As far as biology is concerned, at least this one we can directly take back. And we say that, well, connections, meaning synapses, are responsible. Obviously, we have left out a lot of things, and biology will bring those in determining the nature of the fixed points as well as their locations and how many are there. For instance, we haven't considered here time dynamics, meaning synapses don't have any temporal extent, right? We assume that weight is just W. What happens if you have a weight which is not a fixed number but also changes with time? And in those cases, your nature of the fixed points might change. A stable fixed point might appear as a limit cycle, for instance, and you have transient or persistent oscillations because of that. So a simple answer is that here, as well as in the biology, is the connection weights that are very crucial for Sean.
Yeah, sorry, I'm just going to try to respond directly to the question of this idea of unstable fixed points and whether or not these are real or could be used. I'm thinking about it from the point of view of whether they could be useful. And I mean, certainly we know, and I think this is some of the work that you guys did today, is that the stability of different fixed points can be affected by inputs, right? So you can go from a setting where you have a stable fixed point that becomes unstable as you change some input into a system. And this is a super valuable function, because you could imagine that we know, if we know that we need to act soon, right? We may want to start to crank up the gain on some feature of our dynamics to put us, close to this unstable regime, so that, when we finally have that, moment of the go cue or whatever it is, then we can quickly switch from one kind of activity to another. So I think there is real utility in using unstable or nearly unstable fixed points in the dynamical system in the brain in order to facilitate our behavior.
Yes,
So
Sukbin.
yes, I want to add, to the Arvind and Sean, that the connectivity is also important, and then changing from a stable to the unstable fixed point with the external input is important, but this is under the nonlinear input-output transfer function is only possible. So there is the concept of the effective connectivity, because this input-output gain is changing, depending on the nonlinear is changing depending on the input range. The effective gain is changing. So the effective connectivity, if it is totally linear, then the number of the fixed point would be just a 0, 1, or infinity, or we will not have the changing of this fixed point. But with the combination of the nonlinearity together with the changing effective connectivity, you can have a state dependent, the fixed point, and also it can interact with this connectivity. Yep.
And Sukbin, your work, because you're looking at things like working memory, right? You have to think about stability. It's quite important if you want to hold information over time,
Yes.
that you can push the network into a stable regime. But I guess the question that naturally comes from that is, in order to put something into memory and take it out, you also have to make the system unstable enough that you can, set it up and eventually get rid of that memory. Otherwise, if it was stuck in that perfectly stable fixed point forever, you wouldn't actually be able to remember. So the system does need, in some way, to move into and out of stability, even for something like this, right?
So as Sean pointed out, there can be multiple attractors, and then depending on the external input, you can escape from this attractor and then jump to the new attractor, which can be another memory state, or it can be the basing, right, of the base state. So it can be the memory state. So there is an interaction not only with the external input is also important, not only the recurrent dynamics, but also I want to point out that this instability has been, the study for the disease like the generation of the epilepsy, or in the special working memory. If I not only think about the working memory about the object and then the different attractor dynamics, but if I want to have some kinds of a special attractor, then the first step, the people, to find out the components of this special working memory, I want to find out what type of connectivity is required, or what type of synaptic dynamics is required. So what the people study is studying the homogeneous state, every population. So the population is showing the uniform steady state. And then the people change the, study the bifurcation, where it goes through the instability, and try to find out how the instability arises to account for the this emergence of the special pattern. So this instability is important. Not only for the this disease state, but also to reveal the mechanism of the this generation of some special pattern.
Thank you. So let's move on to.
Well, can I just add one little thing here? That
sure.
When we think of stable and unstable, we should not think that unstable means that the system will explode or die out. It simply means that it can move from one fixed point to another, and that another fixed point itself could result in a bounded input-output relationship. So it's not that you lose your fixed point and you become epileptic, or you just die.
Yeah.
That's a really good point. I mean, I know you want to move on, Eric, but if I could just add one thing, I think this conversation and this idea, and I really like the way Sukbin said it, to remind us that it's really this interplay between the nonlinearities and then the attractor geometry that allows you to have this kind of the kind of movements between fixed points. And that is computation, right? That's what computation is in dynamics. And it's really, from my point of view, one of, if not the most interesting question in the field is sort of how much and how rich can you do things when you're dealing with the interplay between a richly connected network with a complex geometry landscape in terms of its activity and nonlinearities, and what happens when those things come together, and that's what computation is in the context of a dynamical system. And so, I just think this is really, really deep and profound and interesting. And so, we're looking today at these sort of very simple models of one excitatory neuron and one inhibitory neuron, but these concepts start to become building blocks upon which you could potentially construct a human brain. And that's, I think, really pretty exciting.
No, I think that's one of the things that to me is, so just to make a connection to what we were talking about yesterday morning, we had a conversation about generally excitatory inhibitory balance in the brain and why it's important. And obviously, if you had perfect balance all the time, the system was completely stable, you couldn't do any computations, right? We need to be unstable in some sense. We need to be dynamic in order for the brain to actually compute things. And the question is how you set up the network, how you set up the weights and the transfer function such that it has the right properties so that it doesn't, grow unbounded and have like an epileptic seizure, but that it actually does what you want it to do in a way that is both unstable and stable in some sense at the same time. And there are two questions that I noticed that we should just answer quickly, and then I want to move back to a sort of more philosophical question. So the sort of more technical questions, but they came up twice here. So how do we decide what that nonlinearity is? How do we think about the FI curve? Why choose one? And what are the properties that we should consider? So Sukbin, do you want to take a start at that?
So, I mean, as I mentioned, I take a most simplest approach mostly. So I try with the linear model first, and then I try to add nonlinearity. And then even in the nonlinearity, the threshold nonlinearity, whether it can account for the phenomena I'm considering. And then another approach is the utilizing the nonlinear function inspired by the biological, the experimental data. So the experimental data is emphasizing these two types of nonlinearity, the threshold nonlinearity and then saturation nonlinearity. Although in the most of the dynamic range, the people, I mean, the final rate that we are observing, the saturating nonlinearity is not achieved mostly. So maybe with this threshold nonlinearity, I mean, this electrified linear would be simple enough to capture that. But due to this kink, the people made the smooth transition, or like the exponential growth in the beginning, as mentioned in, probably mentioned in the Ken Miller's one. I mean, about the supra threshold, the supralinear suppression. I forgot the name exactly. But then, yeah, SSN, yeah. So for that, like this exponential growth has been utilized, I think, and or the power function, yes. So this combination, I mean, that there are a few choices that people have used in the field. So I would start with this simple nonlinear function.
But you're a mathematician,
yes,
and so my own experience working with mathematicians is that you want simplicity and something that relates to the biology, but is it not also true that you like to choose a form for this nonlinearity that is mathematically tractable, that you you can easily differentiate or that has nice properties. So I'm curious because part of actually thinking about how to model these systems is knowing when it's appropriate to make a simplification to say we'll use an exponential or to use a sigmoidal, or actually I have to use something that's more complex, that's maybe closer to what I've observed in a biological experiment, but becomes less mathematically tractable. So how do you think about making that trade-off?
I'm not sure about the trade-off by, I mean, of course, by the entirely linear versus like this nonlinearity, the threshold nonlinearity is a big change. I mean, about the fixed points, but not about the dynamic property of the fixed points very much. But then, yes, how much it will be qualitatively different, I mean, by choosing the threshold nonlinearity versus more smooth, I mean, the exponential rise with the linear function above, the qualitatively, I don't know how much they will be different. I mean,
well, I was thinking if, you know, you have a point nonlinearity, then, for example, if you want to observe the dynamics as you approach that, you know, point nonlinearity, the dynamics won't be continuous. You know, your ability to do analyses won't be continuous along that point. Whereas if you choose one of these smooth functions, then you have the advantage that you can really look carefully at the dynamics along each of those points in a continuous way.
I think, I mean, that there can be various advantage, I mean, the pros and cons for each type of nonlinearity. So for instance, this rectified linear one, there is some semi-analytical approach by a mathematician actually to try to find out analytically where the fixed points are. I mean, compared to this smooth, I mean, the one, the Karina Cortu's group, not the Karina Cortu, the one in the, do you remember her name? I mean, she gave a talk at the cosine. So she used this rectified linear and tried to identify the fixed point. So, so in some cases, the rectified linear would be better in some sense. I mean, to locate this fixed point or to analyze this fixed point, because you can divide into the two groups, like divide into the two groups where it resides on this threshold nonlinearity or in the dynamic range. And then maybe the fixed point, I mean, this stability analysis would be easier somehow, right? So in some cases, computationally, this rectified linear would be more advantageous. On the other hand, as Eric mentioned, due to this smoothness, right? So without this kinked one, then you may have some, what would be the advantage of this smooth one? Yes, so the smooth one is the shape, the point, the fixed point wise. If you have so this exponential rise one, it can generate some kinds of a rich dynamics by its property itself, or it can also have some different form of the fixed point, not the different form. What I'm saying is, so one of the study by Nicola Brunel and the question in the working memory, I was thinking about the special idea. So one of the question in the persistent activity is that the level of the persistent activity utilizing the typical form of the nonlinearity, then like this threshold nonlinearity, for instance, or like a sigmoidal function, then what you will have as the fixed point is too high, like the persistent activity level, it can be too high. Then he tried to incorporate this exponential rise to explain that the persistent activity observed in the experiment is not that different from this baseline. So the special choice of this nonlinearity can explain some phenomena of the observed in the experiments, or this exponential growth is related to the log normal property. I mean, if you have a Gaussian distribution and then like an exponential one, then you will have a log normal property. So what I'm saying is, depending on the type of nonlinearity, there can be in some case, it's easier to identify the this instability or the stability, or in the other cases, it can be also connected to the other type of experimental observation.
Okay. Sean, I wanted to ask, because a student asked here, when we think about this kind of simple linear or rate model dynamics, or with a simple nonlinearity, as Sukbin was just describing, is this a model of a neuron, or is this a model of a population? We could think about this as basically taking a set of neurons and thinking about their average firing rates. The neurons are homogeneous, with some small variation, or we could think about it as a simplified model of a single neuron. And there was a question here asking, in this case, does it make sense to model the neuron dynamics instead of the population dynamics? But I think we haven't even yet identified how to think about these models. Are these models of populations, or are these models simplified models of neurons? What's the right way to think about that question?
Yeah, maybe instead of answering the question, I'll actually add another option to your list, which is, they may be certain dimensions in the space of the population of the activity of the neurons, right? So rather than a group of neurons, they may be some weighted sum of all the neurons in some potentially complicated weighted sum. And I think that gets to the question of how important is it for us to, I guess, let me take a step back. I think this, we were talking this at the very beginning. The right level of description depends on the question that you're trying to answer, right? And it may be that some, thinking about a rate unit as a population of neurons, or as a linear combination of a bunch of neurons, is exactly the right thing for some questions, but it may not be the right thing for other questions. And in fact, if you don't mind me bootstrapping off of this question back to the conversation you and Sukbin were just having, what I was thinking about with this idea of, well, when do you want to use a smooth transfer function? When do you want to use a linear rectified? When do you want to just assume it's linear? I mean, all of these things, again, are what kind of insight about the network computations and hopefully about brain dynamics can we get? And if we use this particular transfer function, are we able to answer a question that would have been harder for us to answer if we didn't use it? And then I think that it's kind of the proof is in the pudding philosophy of science in a way. So if you get a result, and it's an interesting result, and it gives you some insight about how brains work, then then your choice of the nonlinearity was a good one. I think hopefully then you go back to the experimental data or your experimental colleagues, and they can probe around and they can find something to back it up. But yeah. So I don't know if I helped or hurt in the answer with this question here.
No, I think that was
I
good.
add something. Maybe it's a politically incorrect thing to say, but I don't mind saying those things. So when in doubt, use the simplest form. And if you really don't know what to choose, then choose something that you can handle analytically. For instance, exponential. At least there is one experimentalist who will find your chosen nonlinearity. Okay, this last part is the politically incorrect one. No, I mean, I'm serious now on this, because I think biology is really complicated. Depending on how you experiment with it, you can expose different parts of it, right? So if you think that for your function, you need exponential or log normal or some power law kind of nonlinearity, go for it, try to explain, and wait for the experimental verification of it.
I mean, I think this is an interesting comment. So, often yesterday and today, we're really looking into these biological models that are supposed to allow us to simulate dynamics of parts of the brain. And unlike most of the work we were doing in weeks 1 and 2, where we were taking data from behavior or brains and trying to analyze their activity and look at the results of an experimental analysis, here we're we're presumably trying to model phenomena that have been observed, but we also are trying to generate hypotheses, right? And so I think it's not unreasonable. I don't think you are being unfair to say, if you choose something that maybe you've chosen for convenience, but it then exposes a set of dynamics or a set of behaviors that you think are functionally relevant for the brain and seem to have the properties that you would expect for computation you're interested in, it's totally reasonable to then try to ask, has the brain done something actually that was taking advantage of this? And do we see signatures that look like that? But now I'm going to put you on the spot. Because
Eric, one more. I need to say something else also. Sorry. I think it's relevant here because this question, I think, is very interesting, that what is this equation? Is it an equation of a neuron? Is it an equation of a network? And this is another example where, for instance, this was started by Wilson and Cowan, and they went ahead and they made this bold decision that, hey, we are going to coarse grain it. Turns out the equation is pretty much the same as you would use for the neuron then. But obviously, this is now subject to the constraint that we are assuming that my system is homogeneous, everybody is alike, therefore I can approximate the whole network with one neuron. So in that context, this is a right approximation, and it's justified to call this equation as the equation of the network. This is a mean field description. But if you are now interested in many different types of neuron, your network has heterogeneities in connectivity and neuron types, obviously this is not correct. Then this is equation of a neuron. So yeah, every model has its boundaries, so we should respect those.
So there's a question here that I want to now ask you, because you were saying that the connection weights are the most important thing, right, in thinking about the dynamics of the system. So that was an answer you gave earlier. There's a question here from Leandro asking, how do we know what W is in a real network? And can we estimate it? And I guess this sort of is a question philosophically about the models, but it's also a biological question. How do you relate this W that's so important for the dynamics of your system to what you would go and look for in the biology?
So this, I think, is a very tricky one, both from theoretical perspective, how to define a connection strength. So in these models, we've just put a number. If you have a kernel, you perhaps will say that I'm going to take the area under the curve. If you ask experimentalists, they struggle a lot. What do we mean when we say connection weight? It changes depending on at what level your neuron is, because driving force will change. It changes depending on whether your synapses are plastic, they show short-term dynamics or not. So this number is really very tricky to get. And this is where, at least I believe that this is where I think computational neuroscience suffers, and we are only making qualitative predictions and not quantitative predictions, because this number is not available to us. Experimentally, to measure it, like, what do we mean by this? Typically, we will get a distribution of connection weights. Then what do we do with that? So either when you are doing a simulation, then you can take exactly all those values, the full distribution. Otherwise, you will have to take some movement of this distribution, whether you take care of the mean or the variance or some other movements. There are two hacks that one can use for this to choose your connection weight. At least we know from the network dynamics perspective that the networks behave in qualitatively different regimes when you are working with weak synapses or strong synapses. So this decision you can definitely make, whether you want to study a phenomenon which arises because of weak connectivity or because of strong connectivity.
So I guess.
And what do we mean by weak and strong then? This is also an interesting open question, right? So I think the mathematical definition is for weak connection is that as your network scales, your connection weights, they go down as 1 over square root n. For a practical purpose, because you will never work with infinite synapses, weak means that you need, let's say, some order of magnitude neurons to make your neuron spike. So you can choose 100, you can choose 1,000. If you choose one neuron making another neuron spike, that's a qualitatively different dynamics. So you have to make this decision in terms of how many inputs do you require to make a neuron spike. And based on that, it's a continuum, right? It's not a binary choice. So.
Yeah, so I mean, it sounds though like one of the ways that we can think about this problem is there's not a one-to-one mapping, right? The connection weights that we're thinking about in these more abstracted models, they are capturing many things. They're capturing the structural connectivity, the functional connectivity, the synaptic dynamics, the distribution of weights, across this population, if it's modeling a population.
If I may inject a little bit of controversy, I think
always.
there's been a huge effort in the community, both financial and in terms of person hours, to get connectomes, right? Get connectomes from brains. And some of these are starting to come online, collaborations between Alan and Andreas Tolius and people at Princeton and so forth. And I think there's I personally, at least, am waiting with bated breath, right? Will we find out that there are, when you simulate models with these statistics, is there something interesting compared to what we've done so far? And I think many people, maybe the majority of people, are going to say no. The statistics are not going to be that interesting, and we're not going to learn that much from the connectome. But it's a real. I mean, I think it's worth the effort. I applaud the incredible, hard, and careful work that's going into getting a connectome. So I don't know what Arvind and Sukbin and Eric, what you guys think about this.
I don't know, Sukbin, do you think that like having access to a sampled connectome is going to be helpful for the work you do? What's your guess? She doesn't want to answer for fear.
You have to say it here on format.
Yeah, it's not like this is recorded or going to be available
online. Yeah.
Well, I guess I'll speak for me. I mean, I want to say that I think always information can be useful, right? And so having it obviously is something that we would want to have. I worry in this particular case because we in science, we have a resource allocation question always, and we have to decide where we're going to place our efforts. And sometimes I'm skeptical about where we decide to do that. So I don't know whether, I will try to be not too controversial, but there was a large project funded by the European Union, which was making some pretty strong claims. And those claims involved that we would be able to, replace doing sort of biological neuroscience research using simulations built from ground up models that, and by ground up models, I don't mean models that have been ground up in a grinder, but models that are being built up from low level modeling, where we're modeling the details of compartmental models in complex circuits, and that we would then be able to use those to calculate our more abstract models and verify them and so on. The problem that I saw here is that, I think it was you who said this yesterday, Arvind, if you want to capture all of the details of the brain, the most efficient way to capture all of the details of the brain is to use the brain. And one of the problems with making models is we always throw away things and we don't have some information to constrain the models. And so the idea of moving to this incredibly rich, complex, extremely realistic model to try to answer the questions that we have at this moment in neuroscience seemed to me the wrong time to try to, put the efforts in there, especially when it might take away from the sort of broad areas in neuroscience where we still have many, many open questions. So I think my skepticism, if there's a skepticism about the connectome results and whether they will be immediately useful to us, is are we ready yet to take advantage of them, at least at the scale of even, say, the mouse brain? And the reason I'm skeptical is I have looked at the fully known wiring diagram of C. elegans, which has been useful, but hasn't yet to me sort of transformed what we know about the nervous system of C. elegans and how it controls dynamics in the system and behavior. But that's my take. I don't know whether.
I mean, connectome, of course, is a very nice concept, right? We need the connectivity matrices of the brains. But we don't need it in that excruciating detail. What we need is principles. We need the architecture. whether this is the right way to get the architecture, that you sample every single synapse in the brain and from there derive that principle, or you do old school anatomy and do tracers and so on. Well, so I think that's a matter of choice, like how you are going to answer this question. As theoreticians, we don't need the connectome, we need the architecture. Next we need is that what makes that architecture dynamic, because connectome is not fixed. Our connectivity keeps changing, not just because of plasticity, but physiology of the brain.
No, and I think this is one of the things where there's no right answer, and I think I'm glad, Sean, actually you threw down on this side, because I think it's the sort of thing that oftentimes is dismissed.
Which side did I throw down on? I thought
well,
I just and didn't.
Yeah, I was only I'm trying to push you into a corner by at least you brought it up. I think it's a really important discussion that we need to have, because I think there's always more information we need, and one of the things that I hope in Neuromatch Academy you've been hearing is that, you know, the choice of where to invest your energy in doing analyses or in doing experiments has to in some sense relate to the questions you have and the open space for those questions, right? And I mean, an open and interesting question is sort of, what is the microstructure of the connectivity, say in cortex? And for people who are working on that, you know, the statistics of the connections, to the extent that you can observe them with the techniques we're using, is actually going to be potentially valuable. And you know, we'll just have to wait and see what happens. But I want to get back to a couple of questions that are interrelated here to finish out, because we have we started a little bit late, so we maybe 10 minutes or so left. And there's a question about oscillatory dynamics in these networks, and what creates them, and you know, what is the properties, or why might we think about them, and maybe also how they might relate to the oscillatory dynamics that we can measure, say in ECOG or EEG. So I wanted to start there, and then we'll go from there. So Sukbin, do you want to start by talking about?
So what was the question exactly? The oscillation? What is about the oscillation? And
well, so there are a couple of questions that have been asking about oscillatory models, and so there's a question here saying about how using oscillatory models, what's the relationship to imaginary eigenvalues, for example, when you're doing analyses. And then I want to go on, but the next question will be how we might think about this relating to brain disease. But just first, let's explain and think a little bit about how we study the oscillatory dynamics and what sets them up in these systems.
So oscillatory dynamics, I mean, to understand the principle, I mean, I love from my PhD and my dynamical systems, the first principle is it requires a stronger positive feedback, and then it requires a slow negative feedback. These two constraints are important to generate the oscillatory dynamics in a mechanism-wise. So that means you need a recurrency, right? It is all based on the feedback. So the strength of the positive feedback and then the time dynamics both are important to explain this oscillatory dynamics. So for instance, like the binocular library, there are many, I mean, not only the disease, there are many phenomena based on this oscillatory model, like the binocular library, like there is a perceptual library between the two, I mean, the competing, I mean, the stimuli, and you want to choose one over the others, right? Those kinds of phenomena have been widely, I mean, it's based on these kinds of a rate model, like it requires a recurrent interaction, and also it requires a slow negative feedback. And to explain the slow negative feedback, I think Nicola could have mentioned about the generation of the oscillation in the inhibitory population. I mean, that's the starting point. You may consider like only with the inhibitory population with some kinds of a delay to generate some kinds of a slow negative feedback together. That is a starting point. But if you are having two populations and then like some kinds of oscillation between these two populations, then now you are thinking about the two excitatory neurons, but then you require some kinds of a negative feedback, slow negative feedback with a slow adaptation or spike adaptation or the short-term depression or so on. So mechanism-wise, you can generate the oscillation based on this simple description. Then the brain-wise, the model-wise, the binocular library or attentional change, the people suggest that is based on some kinds of theta oscillation and so on. And how to generate such a theta oscillation? Is it all from the hippocampus, or it can be generated from the local population, or is it from the interaction? So there can be many questions whether this oscillation, so the mechanism wise, either is implemented in the single level or it is implemented in the network. The principle is the same, but the realization can be different. And then like the other question is the oscillation. I mean, the hippocampal one, the people say that is from the single neuron physiology. But then like the other people say that no, it's, I mean, the rhythm is generated with the local network property, or like it's an interaction between the thalamus or the cortex. So there are multiple levels of the oscillatory model based on the different components.
So sorry, Sean, go on.
Sorry, if you don't mind, I was actually thinking about your answer, Sukbin, and it sort of made me think that there might be a nice didactic way to think about this. Earlier, we talked about the importance of the interaction between the nonlinearity and the dynamics, and this is maybe a great example. So there's a question here from Maya about the role of imaginatory eigenvalues, and of course, in a purely linear system, you can have oscillations from imaginary eigenvalues. But you can also have a nonlinear system where if you linearized it at any point, and it's a nonlinear system, let me repeat what Sukbin said, which is that has fast positive feedback and slow negative feedback. And if you linearized it, in the phase of the fast positive feedback, you would see that the eigenvalues of that phase are positive. And then when that negative feedback is really kicking in, you'd see that the eigenvalues of the linear system of the Jacobian at that moment are negative. And so those are a sort of a switch in the dynamics of the real part of the eigenvalue that's given through the nonlinearity, as opposed to the oscillations that are intrinsic to the linear dynamics that are given by the imaginary eigenvalue. So there's sort of two different kinds of oscillations that you can have, whether it's a consequence of the nonlinearity or if it's a consequence of the connectivity, which dictates the eigenspectrum. I don't know if that's helpful for anybody, but I think it's interesting.
No, I think that's a really, so I was going to try to put together this concept of whether the time constant, the fundamental difference in the time constants and feedback is the only way to think about it. And I think, you just pointed out that this can actually be driven sort of by two components. What I'm curious about is one of the questions here was, can you get this only from this kind of recurrent dynamics, or do we see in the brain places where this oscillation might be driven externally? And I guess here the question, and it relates to what Sukbin was saying, is what is external? So if we're looking at two populations and say an area in the visual system that we think are involved in binocular rivalry, we might be looking at the internal dynamics, the competition between these things. But it could be that if we're observing them, at least in the biological space, that, oscillatory activity is being driven from somewhere else, as an external oscillatory input, like from the hippocampus. And I don't know exactly what the question was going to drive at, but is there a way we can tell those apart if you're just doing an observation of the system? This is a philosophical question.
Whether it is internally generated or it is driven by the external input, that's what you're saying? How can it distinguish, this oscillatory activity? I don't know. If anyone has some answer for that, the perturbation would be the perturbation study, whether you will suppress all of this, the oscillatory activity or not by, the silencing some of this. partial, the local activity. Yes, but there is
yeah, that would
also
be if you could, for example, if you could silence the feedback connections. Then you would, if you still saw those oscillatory dynamics, then you could say perhaps they're not, they're being externally driven because we think we've silenced the local recurrent feedbacks, which perhaps we could do through an optogenetic manipulation. I mean, it's very easy to do in the model. You simply turn down the weights between the E and I populations, for example, right?
Yes, the perturbation was one of the answers I provided, but I want to give you some warning to interpret the perturbation one as well. Some of the study I have investigated was like the working memory model, but then although the persistent activity was generated, there is all mechanism in the local circuit, but there can be some of the mechanism which can be robust against some type of a perturbation. Okay, so so one of the requirements to generate this persistent activity, I investigated was the balance between the E and I, Eric mentioned. And then the perturbation type, for instance, the silencing some part of the activity, this local activity, actually it was shown that it doesn't disrupt the E and I balance. So I want to be careful. The perturbation study you may consider, to find out the locus of some generating some activity. But on the other hand, when you interpret that, there can be some kinds of a compensatory mechanism or what type of perturbation you are considering. You have to be careful when you interpret. So even if it doesn't disrupt this activity in this local circuit, with some special type of perturbation, you cannot preclude, exclude all the possibility that it may be still generated in the circuit. Yes, I'm just generating more confusion, maybe. Yes.
So it's a good. I think that was a good answer. I think the other thing that I can think of as an example of external drive is that we know that if you have potential oscillatory dynamics in a system, then you can also entrain those dynamics if you provide an external input that has the same time constant, right? That you'll get positive feedback in the system. So like, I'm thinking of the case where if you put a strobe light in front of somebody, right, you'll generate waves of activity in visual cortex, and those waves of activity, if they're at the right frequency, they can actually drive the system right into a different regime. And in some cases, people can have epileptic activity, for example, or you could also get entrainment, right, where you get actually local oscillatory activity that is being driven and gets amplified by the time constants, in the network. So at least that's another way that we might have external drive that causes oscillatory activity, real external drive. And that, I guess, brings us to the questions I wanted to get to about how these kinds of models might relate to brain diseases and dysfunction. Because clearly, changing the time constants within these network models can create very different regimes, some of which are very unstable, or some of which get locked into oscillations and no longer will be doing the kinds of computations we think are going to be valuable for the system. So I don't know, Sean, do you want to say some things about how we can think about these models as a way of starting to think about the connection between computations that are normal and dysfunctional, and specifically maybe because people are interested with respect to oscillations or the stability of these kinds of dynamics?
Well, first I want to caveat it by saying that I have no special knowledge here at all. But I think that the connection is pretty straightforward. I mean, we know that epilepsy is a runaway oscillation state, right? And there are, one of the things that's important to recognize is that epilepsy is many different things. It's not one thing. And in fact, it may be as many things as there are patients who have it to some extent. But certainly it is also a phenomena of uncontrolled oscillation. And so thinking about how you go from a dynamical regime where you're able to have complex asynchronous activity in the brain to a regime where you suddenly have massive synchrony is of incredible importance, not only to understand and treat epilepsy, but I think, frankly, to sort of help us think about how the brain does the functions that it does. You know, how does it stay in its stable regime most of the time for most of us? In fact, runaway oscillations, maybe many of our students have played with, or at some point in their careers will play with, recurrent neural network models. I play with them all the time. It's really, really easy to get those things to oscillate. It's actually kind of hard to get them to not oscillate. So, so that's interesting, right? How, you know, what are the brakes on the system that prevent that, and then how can they become disorganized? I don't know if I had anything more, if I have anything more intelligent to say than that.
No, I mean, I think that was a really good start, and I think, I mean, one of the questions, maybe I don't know whether you want to speculate or one of the other panelists, but what's the space, right, in the parameter space, let's say, of these networks, where you get the right balance between stability and instability? How much of the space, the parameter space that we might be able to play with, and here we have the weight matrix and the FI curve and so on, or the time constants of the two different populations. There's a number of parameters, and the bigger and more complex the network, the more they are. How many, you know, how much of that space is going to lead, as you said, to producing dysfunction, either no activity or locked, you know, oscillatory activity you can't escape from, or explosive activity? And how much of it is in the regime where the system really does the kinds of computations that we're interested in?
Yeah, and how much of learning is actually geared rather than to consolidate knowledge, but to prevent those parameters from getting into maladaptive regimes? I mean, that's another interesting question.
Arvind, do you have anything you want to add to this?
So specifically, what do you want me to add on?
Well, I'm curious if you want to think, if you wanted to provide any further thoughts on the connections between sort of how we can use these kinds of models to think about normal function and dysfunction in the system, and maybe stepping away just from the case of oscillations, but like what are the ways that we can think about what happens as you move out of the regime, which you think is doing something that's
I think I can say a lot, because at least personally, I believe that a lot of brain diseases are actually diseases of brain dynamics. Epilepsy is obviously a classic example of a system being unstable. But for instance, Parkinson's disease is yet another example where your system don't necessarily enter into epilepsy, but it leaves its stable fixed point of non-oscillatory activity and moves to an oscillatory regime. So there are several examples of this sort that you can find in like this large-scale behavior, right? Like Parkinson's disease or epilepsy. To some extent, you can relate this to Alzheimer's disease, where you lose your memory representations. If they are as attractors, then this would fit very well. That as you sparsify the network, you lose connections, you lose your fixed points, and therefore you lose your memory or retrieval of memory. So I think these kind of models are extremely helpful in creating a nice conceptual framework to think about diseases beyond molecules. I mean, I can really say it without worrying. It's a really disgrace, you know, that when we think of brain diseases, we talk about a brain function and then immediately jump to the lowest level possible at the molecules and the genes. And we leave these 12 orders of magnitude in between, and we never worry about it, at least not as a clinical scientist. And I think that's where theoreticians could come in and create a bridge between these different levels. So that's super important. I also want to weave in now this one question that I really like. It's by Andy, that it says that maybe a few words about how to model the brain in a statistical or information processing way. So there are certain diseases that you wouldn't call them as diseases of dynamics, but perhaps diseases of information processing. Like brain of a schizophrenic patient, dynamically speaking, I think it's perfectly normal. It has its fixed points, it can switch between different dynamics and so on. The problem perhaps is that the way this person, or we use Bayesian framework to understand it theoretically, that there is an imbalance between, let's say, top down and bottom up. If you are too much in top down, then perhaps you will end up being schizophrenic. If you are too much bottom up, most likely you will end up as ADHD. So this is another way to think of the brain function as networks, that it's an information processing device. It needs to infer something about the environment. So that's yet another way of modeling the brain besides this dynamical systems approach. Here we are behaving like physicists. When we think of the brain as an information processing system, we behave like we are computer scientists or statisticians, that we have to infer the state of the environment through whatever priors and other sensory evidences are there. So that's another way to think of models of the brain function. A third approach I kind of like is also coming from the observation at the neuron level, which is taking the advantage of tuning curves. So you have neurons which are specifically tuned to respond to certain type of stimuli or features. You can put them together and create what is called neural engineering framework to build networks which do information processing the way you want it to be. So there are several other approaches to think about brain's networks. And what we did today is just one way to think about the dynamical properties. And as I was earlier saying, that these dynamical properties do relate to brain's function and dysfunction.
Yeah, I think that's, it's really important that we often jump from, concepts of disease to say something that we think might be a change in the genome that, leads to one misfolded protein. But in fact, that may or may not be the right way to start at all. In fact, thinking about it at a level that's more related to how we diagnose and think about the change in properties of the system. So that's behavior and representations is important. But Sean, you wanted to say something. So what were you?
I was just really excited by what Arvind was saying. And the one thing that I wanted to quibble on a little bit was this idea that say something like schizophrenia might be better modeled by thinking about the relative balance of top up and top down information in the network. And that makes perfect sense. And I agree with that. But I think that the problem with a purely Bayesian modeling approach there is that ultimately those functions are implemented by network dynamics. And that's where there's a real question. And frankly, so I don't know if the folks here know, but I'm trained as a psychiatrist in addition to being a neuroscientist. And I don't work on this because I don't know how to start, but my my number one question that I would love to be able to try to get an answer to is exactly that one: is how do the dynamics in neural networks give rise to the functions that could become disorganized in an inference machine like in schizophrenia? I mean, that's something that I'm really interested in.
And this is a good place to maybe wrap up. By pointing out that this is an enormous set of open questions. So there's a whole field that's begun recently called computational psychiatry, which is really trying to think about ways of using computational modeling approaches to relate function and dysfunction at a level that is in between where we have often looked, with pharmacological access to particular sites in neurons, particular receptors, or gene dysregulation, and on the other end, just symptomatic description. So if we have models of dynamics or functional properties, and even if they're extremely simple, and Sean will tell you, most of this information is just missing right now, there's a whole new field that is growing up to try to start thinking about this. And so it's a good opportunity if you're interested. And I'm going to make one small side note, because there's a question here about glial cells, the most unloved cells in the brain, which actually comprise an enormous fraction of your brain, far more than you probably realize. And they are, of course, doing extremely important things. They're helping to create homeostasis. And there are links that we believe exist potentially between their function and disease. But one of the interesting things is I think we know very little about how to add them to our models right now. So we know that some glial cells have active properties. We know that they're involved in regulation because they regulate glucose access and they take up neurotransmitters, so that they're going to be involved in the sort of steady state dynamics of these systems, but we leave them out of every single model. So there's another area that you can start investigating. So with that, I guess, do you all want to make any last comments? Was there anything that you'd like to leave the students with? I think we've answered a good number of
Can I say something?
questions.
Some advice that I received once.
Sure.
It's very tempting to put a lot of biological detail, and then you will realize that you can't do anything analytically, so you will go to simulations. Simulations are very powerful. I use them all the time. But the most important advice I received about simulations was never simulate anything until you know what to expect. And what to expect, that intuition you can get from these simple models, at least to some extent.
Or from perhaps looking through what we've observed in biology, hopefully. And with that, I want to thank Sukbin and Sean and Arvind for joining us. And I hope you've enjoyed this conversation. I hope we've answered at least some of your questions. And please continue the discussion on Neurostars. If you have questions that are technical on details of the tutorial, there'll be many people who will answer them for you there. And we look forward to seeing you again tomorrow. So thank you all, and we'll see you soon.
Yeah, thank you, Eric. Thanks, Sean.
Thanks,
Bye.
thanks, Eric.
Bye-bye.
Thanks, everyone else. Take care.